Welcome to our Introduction to Sterilization, a fundamental process in microbiology and healthcare. Sterilization is defined as the complete elimination of all forms of microbial life. This includes bacteria, viruses, fungi, and even resistant bacterial spores. Sterilization is critically important across multiple fields. In healthcare, it ensures surgical instruments and medical devices are safe for use. In microbiology laboratories, it prevents contamination of cultures. In research, it ensures experimental integrity, while in industry, it's vital for pharmaceuticals and food safety. It's important to distinguish sterilization from related processes. Unlike disinfection, which reduces microorganisms to safe levels, sterilization completely eliminates all microbial life. Sanitization reduces microbes to acceptable levels, while antisepsis targets microbes on living tissue. Proper sterilization is essential for preventing infections and contamination. It protects patients during medical procedures, ensures research validity, and maintains quality in industrial processes. Failure to properly sterilize can lead to serious infections, contamination, and compromised scientific results. Now that we understand what sterilization is and why it's important, let's explore the key terminology used in this field. Understanding the key terminology in sterilization is essential for proper infection control. Sterilization refers to the complete elimination of all microorganisms, including bacteria, fungi, viruses, and spores. Disinfection eliminates most pathogenic microorganisms, but typically doesn't affect bacterial spores. Antiseptics are chemical agents specifically designed for use on living tissues to prevent infection by inhibiting microbial growth. Asepsis refers to the practices and procedures that prevent contamination from pathogens, creating and maintaining a pathogen-free environment. These terms are closely related and form a hierarchy of infection control measures. Asepsis encompasses all techniques for preventing contamination. Within that, disinfection and antiseptics are methods to reduce microbial presence. Sterilization represents the most complete form of microbial elimination. Let's examine when each approach is appropriate in different healthcare settings. Sterilization is essential for critical items entering sterile body tissues, like surgical instruments. Disinfection is suitable for non-critical items that contact intact skin. Antiseptics are used on living tissues requiring microbial reduction. Aseptic techniques create and maintain sterile environments for procedures. Understanding these key terms is fundamental to infection control practices across all healthcare settings. Physical sterilization using heat methods is one of the most effective and widely used approaches for eliminating microorganisms. Heat effectively destroys microorganisms by denaturing proteins and disrupting cell membranes. Heat sterilization methods can be divided into two main categories: dry heat and moist heat. Dry heat methods include direct flaming, hot air ovens operating at 160 to 180 degrees Celsius, and incineration. These methods work primarily through oxidation of cell components. Moist heat methods include boiling at 100 degrees Celsius, steam under pressure, and autoclaving at 121 degrees Celsius for 15 to 20 minutes. These methods work by denaturing and coagulating proteins. Autoclaving is considered the gold standard for most laboratory equipment sterilization. An autoclave uses pressurized steam to reach temperatures of 121 degrees Celsius, which is more effective than boiling water. Autoclaves typically operate at 121 degrees Celsius and 15 pounds per square inch of pressure for 15 to 20 minutes. Proper loading of an autoclave is crucial for effective sterilization. Items should be arranged with sufficient space between them to allow steam circulation. Different heat sterilization methods are appropriate for different applications. Autoclaving is ideal for laboratory equipment and media, hot air ovens for glassware and metal instruments, and direct flaming for inoculating loops and needles. Heat sterilization remains one of the most reliable methods for eliminating microorganisms in laboratory and healthcare settings. This section covers physical sterilization methods using radiation and filtration. Radiation sterilization uses ultraviolet light and gamma rays to eliminate microorganisms. Ultraviolet radiation, particularly UVC, with wavelengths between 200 and 280 nanometers, is highly effective for surface sterilization. UV light works by damaging microbial DNA. It causes thymine dimers to form, which prevents DNA replication and leads to cell death. Gamma radiation uses high-energy electromagnetic waves from isotope sources like cobalt-60 or cesium-137. Gamma sterilization offers significant advantages for heat-sensitive materials, but requires specialized facilities and has some limitations. Both UV and gamma radiation work by damaging microbial DNA, but through slightly different mechanisms. Radiation energy breaks chemical bonds in DNA and creates free radicals that cause additional damage. This damage prevents microorganisms from replicating and ultimately leads to cell death, achieving sterilization. Filtration sterilization physically removes microorganisms from liquids, gases, or air using various types of filters. HEPA filters remove at least 99.97% of particles that are 0.3 microns or larger in diameter. HEPA filters work through a combination of mechanisms including interception, impaction, and diffusion as air flows through the filter medium. Membrane filters have much smaller pore sizes than HEPA filters, typically between 0.1 and 0.45 micrometers, which can remove bacteria and some viruses. Different types of filters have specific applications based on their pore size and the microorganisms they can trap.
Sunlight acts as a natural sterilization method through its ultraviolet radiation component. Sunlight contains different types of UV radiation. UVA has minimal germicidal effects, UVB has moderate sterilization capability, while UVC is most effective but is largely filtered by the Earth's atmosphere. UV radiation damages microbial DNA by creating thymine dimers, which prevent proper DNA replication. This disruption leads to cell death or inability to reproduce. Solar disinfection has several practical applications. SODIS is a water purification method where contaminated water in clear plastic bottles is exposed to sunlight. Sunlight is also used for disinfecting laundry and surfaces in resource-limited settings. Despite its benefits, solar disinfection has limitations. It's highly dependent on weather conditions and cloud cover. Effective sterilization requires at least 6 hours of exposure, and sunlight cannot penetrate opaque materials. Ozone, a triatomic oxygen molecule with the formula O3, is a powerful oxidizing agent used for sterilization. Ozone's sterilizing effect comes from its ability to oxidize cellular components. It damages cell membranes and proteins, disrupts enzymatic systems, and breaks down nucleic acids, ultimately leading to microbial death. Ozone is widely used in water treatment systems to disinfect drinking water and wastewater. It's employed in air purification systems to eliminate odors and microorganisms. Ozone also finds applications in food processing and medical equipment sterilization. Ozone has several limitations. It can be harmful to humans at high concentrations, requiring careful monitoring and containment. It has a short half-life, meaning it must be generated on site. Ozone can also damage certain materials like rubber and plastics. Both sunlight and ozone offer effective physical sterilization methods with specific applications and limitations that must be considered when implementing them in practical settings.
In this section, we'll examine halogen-based disinfectants and phenolic compounds used in chemical sterilization. Halogens include chlorine, iodine, and bromine. These elements are highly effective disinfectants used across various applications. Halogens work through oxidation, a chemical process that disrupts critical cellular components of microorganisms. This oxidation disrupts enzyme function, damages cell membranes, and prevents protein synthesis, effectively killing microorganisms. Chlorine is widely used in water treatment systems, swimming pools, and household bleach. Iodine products like povidone iodine are essential for wound disinfection and surgical site preparation due to their broad-spectrum antimicrobial activity. Now let's transition to another important class of chemical disinfectants, phenolic compounds. Phenolic compounds are derivatives of phenol, which consists of a hydroxyl group attached to a benzene ring. Common derivatives include cresols, chloroxylenols, and bisphenols, each with specific antimicrobial properties. Phenolic compounds disrupt bacterial cells through multiple mechanisms. They penetrate the cell wall, disrupt membrane integrity, denature proteins, and inactivate essential enzymes. Many common commercial disinfectants contain halogens or phenolic compounds. Phenolic products like Lysol, chlorhexidine, and Dettol are widely used in healthcare and household settings for their effective disinfection properties. Halogens are ideal for water disinfection, wound care, and food processing equipment due to their broad antimicrobial effectiveness. Phenolic compounds excel in hospital settings where robust environmental disinfection is crucial, particularly for surfaces and equipment. When using these chemical disinfectants, it's important to consider appropriate contact time, material compatibility, and safety precautions such as ventilation and protective equipment. Both halogen and phenolic compounds provide effective chemical sterilization options for different settings and applications. In this section, we'll examine oxidizing agents and gases used for chemical sterilization. Oxidizing agents are powerful chemical sterilants that generate free radicals to destroy microorganisms. Hydrogen peroxide is a widely used oxidizing agent available in concentrations from 3 to 59%. It's effective against a broad spectrum of microorganisms. Hydrogen peroxide generates hydroxyl free radicals that attack cell membranes, DNA, and proteins of microorganisms. Peracetic acid is a fast-acting oxidizer effective at low temperatures. Unlike other chemical agents, it breaks down into non-toxic acetic acid and oxygen, leaving no harmful residues. Vapor phase sterilants are gases used to sterilize heat-sensitive materials and devices that cannot be sterilized by other methods. Ethylene oxide is a highly penetrating gas used for heat and moisture-sensitive items. It's particularly valuable for sterilizing electronic devices, plastics, and optical equipment. Ethylene oxide is commonly used for sterilizing electronic medical devices, catheters, implantable devices, and other items that cannot withstand high temperatures or moisture. Chemical sterilants come with important safety concerns. Hydrogen peroxide is corrosive to skin and eyes, while ethylene oxide is carcinogenic, mutagenic, and flammable, requiring special handling protocols. Proper ventilation is critical when using gaseous sterilants. Facilities should have dedicated sterilization rooms with negative pressure, frequent air exchanges, and gas monitoring systems to protect healthcare workers. Chemical sterilants like oxidizing agents and gases effectively destroy a wide range of microorganisms, making them valuable tools in healthcare settings. Section 9. Selecting the appropriate sterilization method. 3 key factors influence the selection of an appropriate sterilization method: material properties, microorganism resistance, and specific application requirements. Material properties significantly impact method selection. Heat-sensitive materials like plastics or electronics cannot withstand autoclaving, while moisture-sensitive items may be damaged by steam sterilization. Different microorganisms exhibit varying levels of resistance to sterilization. Bacterial spores are the most resistant, requiring more intensive methods, while vegetative bacteria are relatively easy to eliminate. Application requirements also play a critical role. These include processing time constraints, required sterility assurance levels, batch size, cost considerations, and regulatory requirements specific to different industries. This comparison table highlights the key differences between major sterilization methods. Note that physical methods, shown in teal, include steam, dry heat, and radiation, while chemical methods, in orange, include ethylene oxide and hydrogen peroxide. This decision-making framework provides a simplified approach to sterilization method selection. First, evaluate if the material is heat-sensitive. For heat-sensitive materials, chemical methods like ETO or hydrogen peroxide are appropriate. For heat-resistant materials, steam autoclave offers efficiency and effectiveness. Special considerations may apply in complex scenarios. Some items might require combination methods, while packaging compatibility and environmental impact are increasingly important factors. For example, a complex medical device with electronic components and heat-sensitive polymers would typically be sterilized using ethylene oxide with an extended cycle time. To summarize, selecting the appropriate sterilization method requires carefully matching the material properties, target microorganisms, and specific application requirements. This systematic approach ensures effective sterilization while preserving material integrity. Validation and monitoring are critical final steps in any sterilization process. Without proper verification, we cannot ensure that sterilization was successful. There are three primary verification methods for sterilization. Biological indicators contain resistant bacterial spores and are the gold standard for validation. Chemical indicators like color-changing tapes and strips respond to specific parameters and provide immediate visual verification of exposure to sterilization conditions. Physical monitors measure parameters like temperature, pressure, and time, providing real-time data during the sterilization process. Let's look more closely at biological indicators or spore tests, which are the most reliable method for validating sterilization effectiveness. Spore tests work by following a simple four-step process. First, you place a biological indicator containing highly resistant bacterial spores in the sterilizer along with the regular load. Next, you run a normal sterilization cycle according to your established protocols. After sterilization, you incubate the indicator for 24 to 48 hours to allow any surviving spores to grow. Finally, you observe the indicator for growth or color change, which would indicate that sterilization failed. When interpreting results, a positive test showing growth or color change means sterilization failed and items must be reprocessed. A negative result indicates successful sterilization, confirming that the process effectively killed even the most resistant microorganisms. Quality control procedures ensure consistent and reliable sterilization results. Proper documentation is essential for regulatory compliance and traceability. Documentation should include date, time, and operator identification. You must record the sterilizer identification and all cycle parameters. A description of the items or load being sterilized must be documented. Results from all indicators, biological, chemical, and physical, must be recorded, and any corrective actions taken for failed cycles should be thoroughly documented. Establishing a regular validation schedule is crucial for maintaining sterilization effectiveness. Biological indicators should be used daily to weekly, depending on your facility's requirements and risk level. Chemical indicators should be included with every load to provide immediate verification of exposure. Physical monitors automatically record parameters for every cycle and should be checked consistently. A full qualification of the sterilization equipment should be performed annually or after any major repairs or modifications. Proper validation of sterilization processes serves several critical purposes. First and foremost, it ensures patient safety by preventing infections and protecting vulnerable individuals from harmful microorganisms. In laboratory settings, validation maintains experimental integrity by ensuring reliable research results and preventing contamination of sensitive experiments. Finally, proper validation demonstrates regulatory compliance with accreditation standards and legal requirements, protecting your organization from liability. Remember, without proper validation, sterilization cannot be confirmed, putting patients, experiments, and your organization at risk.